Good day, everyone. Before I begin my report, I would like to first thank you all for watching this video. My goal here is to explain the topic clearly and make sure everyone understands the key points. So, what is genetic basis of disease resistance? The genetic basis of disease resistance refers to how an organism's genes enable it to fight off infections, tolerate pathogens, or prevent diseases from developing. In simple terms, it explains how DNA makes some individuals naturally better at resisting disease than others. How genetics influence resistance to pathogens. So, as we all know, disease resistance refers to an organism's ability to fight off infections, and this ability is strongly influenced by its genetic makeup. It is important in aquaculture breeding because diseases can rapidly spread in high-density fish environments. By studying genetic resistance, breeders can develop stronger, healthier populations and reduce losses due to outbreaks. And it also improves the stock survival of our fish farm. And why disease resistance matters? First is diseases cause major economic loss. Fish farms often raise thousands of fish in one area, making it easy for disease to spread quickly. When an outbreak happens, large numbers of fish can die in a short time. This leads to a huge financial losses because farmers lose their stock, waste food, and must spend more on treatments and disinfecting the facility. And also, the reduction of antibiotic and chemical use. Because if fish are genetically resistant, farmers don't need to rely heavily on antibiotics or chemical to keep them healthy. This is important because too much antibiotic use can lead this to resistant bacteria and harm the environment. Having naturally disease-resistant fish means healthier production with fewer medical interventions. And the third one is it improves sustainability. So, genetically resistant fish survive better. Yes, grow consistently and require fewer resources. This supports a long-term sustainable aquaculture by reducing waste, lowering mortality rates, and promoting eco-friendly practices. So, sustainable production ensures the industry can continue without harming the environment or producing unsafe products. The next is the genetic factors influencing resistance. We have here 4 factors, which is the major genes, polygenic traits, genetic marker for resistance, and genomic selection tools. Some fish have specific major genes that give them strong protection against particular diseases. In most cases, disease resistance is polygenic, meaning many genes contribute small effects. Scientists use genetic markers to locate these resistance traits in the genome. With modern genomic technologies, breeders can easily identify which fish carry beneficial genes and use them for breeding. And the next is the mechanisms of disease resistance. First is the physical barriers, innate immunity responses, adaptive immunity, and the gene expression during infection. The first line of defense is the fish physical barriers, such as skins, scales, and mucus, which prevent pathogens from entering. If these barriers are breached, the innate immune system responds rapidly. Over time, the adaptive immune system forms antibodies and memory cells to fight specific pathogens. Genetics influences how strong each of these responses is, determining how quickly and effectively fish recover from infections. And the next is the selective breeding for disease resistance. We have here the identify resistant broodstock, marker assisted selection, genomic selection, and produces healthier populations. Selective breeding involves choosing fish that naturally survive disease challenges and using them as parents for the next generation. With marker-assisted selection, breeders use DNA markers to identify resistance genes. Genomic selection uses large-scale DNA information to accurately predict which fish will be resistant. These methods create robust fish populations which improves long term. And we have here the case study KHV resistant carp. And it developed through selective breeding, higher survival against koi herpes virus, and genetic screening identifies resistant lines. Carp's populations have been heavily affected by KHV or koi herpes virus. Breeders responded by selecting carp that survived KHV outbreaks and breeding them together. Over generations, this produced carp that are naturally resistant. Genetic screening helps identify which individuals carry resistance traits, ensuring the trait is passed on consistently. This case shows how genetic can solve real industry problems. And the next is applications in aquaculture. Prevents large-scale mortality, reduces chemical treatments, improves productivity, and supports long-term breeding goals. Applying genetic resistance in aquaculture leads to strong disease-resilient populations. Farms experience fewer outbreaks, meaning less reliance on antibiotics or chemicals. This results in higher yields, better fish quality, and improved economic stability. Over time, incorporating disease resistance into breeding programs ensures sustainable and profitable aquaculture operations. This conclusion highlights the importance of genetics in shaping disease-resistant traits. It explains that resistance is not random. It's passed down through genes, meaning breeders can intentionally strengthen this trait through selective breeding by choosing fish that naturally survive disease challenges. Aquaculture operations can enhance population health and reduce the need for chemical treatments. This leads to a long-term sustainability, healthier fish, and better overall production efficiency. And that's all. Thank you. Thank you for watching.